We're so excited to have you here with us at Just Like Nana. Today, I am absolutely thrilled to be interviewing Dr. Isabelle Mansuy. She runs a neuroepigenetics lab as part of the Brain Research Institute at the University Zurich and the Institute for Neuroscience at the Swiss Federal Institute of Technology. She studies generational trauma. Dr. Mansuy's credentials and accolades are too numerous to list. Highlights: she's a member of the Swiss Academy of Medical Science and was elected Knight of the Legion of Honor in France. Welcome.
Yeah, thank you.
Lovely to meet you. Thank you so much for being here.
My pleasure.
As you know, this is a podcast that talks all things, we call it ancestral trauma, but intergenerational trauma, generational trauma. I would love to start like we do with every guest and just hear one of your favorite memories of a grandma or Nana and what you call her. Would love to hear that.
I'm afraid I don't have any special memory with my grandmas because both were somewhat, one was very authoritarian
Ah,
and the other one was sweet, but for a reason. Yeah, I looked for memory, but I don't have anything special.
that's okay. What's your word for grandma?
It depends. One of them was grand-mère, which is very formal because in that family from my mother's side, my mother herself was not saying tu. In French we say tu or vous. This very, very formal way to name someone. They were very formal, even if they were from the countryside. The other grandma, we called her mémère, which means she's really not sweet because mémère is very old, means very old lady, mémère.
I would love to talk about your work. This is a very kind of simplistic question, but we call this ancestral trauma. It's also referred to as intergenerational trauma or generational trauma. I kind of think of intergenerational trauma as more trauma that follows through a family line, and we'll talk about your work in epigenetics. Generational trauma as more kind of a generation of people, so how war, historical events affect that generation. And then ancestral trauma is a little bit more of a human kind of woo and not scientific way to talk about your family and how your family affects who you are. Do you see a difference in those terms? What term do you use?
We don't use the term transgenerational ones, ancestral trauma, because it's not precise enough and it's indeed misleading and wrong because it's not the trauma which is transgenerational, it's consequences. And it's very important because if you have a transgenerational or intergenerational trauma, it means that the trauma itself is reiterated, reproduced each generation, right?
Okay.
And this is not the case. Indeed, what people generally mean is that there was trauma in one of the prior generations, in the ancestors, and that the descendants are subjected to the consequences, to the effects, generally and negative effects of that trauma. So I think it's very important to be more precise and say that I know that it's a shortcut, which may be convenient, but because people understand it somewhat, but it can be very misleading because if, let's say, you take someone, or let's say a man who has been traumatized, abused or whatever when he was a young boy, he himself will be violent with his children and repeat the conditions that made him violent, depressed and antisocial and everything. And then the following generation will again be exposed to exactly the same trauma. So then it's very different from the inheritance where it's really the consequences, because from a biological perspective, we are talking about different things.
Yeah. Okay. That's a great point. And that kind of leads into, can you explain epigenetics? What does it mean? How does it inform your work?
Yeah. Epigenetics is the complement of genetics. So in each single cell in our body, we have the genome, which is the DNA sequence. It's a little bit like the hardware of your computer. And in these cells, on top of the DNA sequence, next to the DNA sequence, there are epigenetic factors, which can be very different. So while there is one code, just one code, made
Yes.
4 different bases, of course, the code is super long and complex, but it's just one code. For the epigenome, there are many different epigenetic marks, which interact, which are very different from cell to cell. And these epigenetic marks, which are on the DNA itself and around the DNA, they modulate the activity of the genome. So it's a little bit like the softwares of the computer. A computer without softwares has the code, but it's useless. You cannot do anything. You need the softwares to read it, interpret it, and so on. And you need multiple softwares. And these softwares, you can add them, remove them, they can interact, and so on. And that's, also that gives the right notion of the level of complexity. While there is one code, the epigenome is extremely complex. One code, and if you damage it, you break it. If your hard drive has a scratch, it's done, right? You cannot fix it. The DNA is big like that. If you have mutations due to X-rays, carcinogens, UVs, they cannot be really fixed. While the epigenome, it's very dynamic. It can be modulated, it can be changed, it can be corrected, repaired. So it's a complement of the genome. It's necessary to its function, but it has another scale, another level, another dimension in its dynamics and reversibility in a way. And the environmental factors, what we experience, what we eat, the sport we have, our emotions, everything outside modulates the epigenome in every single cell of our body, and maybe differently in different cells. If you eat good food, vegetable and fruits, they will act on all your cells, but differently on your brain cells and on your liver cells. If you go for a run, the epigenome will be changed in your body, but differently in muscle cells and in skin cells. So the epigenome, it's another properties. It's different from one cell to the next, while the genome is exactly the same in every single cell of our body.
Okay. And the activation or modulation of what word did you, the epigenome?
The genome is the DNA code.
Right.
The genome, genes, there are genes.
Okay. Yes.
And sequences outside genes. Genes, it's only a very small proportion of the long sequence.
Right.
But all the rest is sequences in between genes. So the epigenome, epigenetic factors, they modulate activity of the genome.
Okay. And then it will occur, getting back to your example of fruits and vegetables and running, it's modulating at the time, but then that also continues to have effects afterward. Is that fair?
Of course, it modulates, the DNA is active all the time. Of course, genes are not expressed all the time, but the DNA itself, there is always something going on around activity around the DNA, and epigenetic factors are the same. They are very dynamic, and the effect can be immediate, but some of these changes in epigenetic factors, they can last, and some of these changes, they can be permanent indeed.
Maybe this is a good time because I have more questions about humans, but can you explain your work?
My work is to study or try to understand what mechanisms operate in the body, which can explain how life experiences, in particular in childhood, can modify our mental and physical health. So we are particularly interested in these factors in germ cells because we want to understand how life experiences can modify people across generations. And here, when we are talking about across generations, we really focus on germline dependent transmission. Because what is the link between a father and his children? Biologically, what is the link? It's the sperm, right? Sperm provides the genome, the genetic luggage, and the epigenetic luggage of the father. to the child. And we are interested in the mechanism at this level. So the biological luggage signals that a father can pass to the children and which carry information about his own life experiences.
And that's interesting because I'm curious about the difference between men and women. So obviously women are born with their ova, what they're going to pass on to their children, use to make their children, and men are consistently reproducing sperm. So is there a difference? Like, are the life experiences of a woman even further embedded, if that makes any sort of sense?
We don't know, but probably not, because, sperm, they don't come. Of course, they are produced all the time, but they come from a progenitor cell. And this progenitor cell is present in the man, in the boy, very early on. So if this progenitor cell is affected by stress, by poor diet or endocrine disruptors or whatever, it's possible that some of the alterations will stay in these progenitor cells, which is going to be produced in the future. We'll have that. Of course, now if we take an adult man who gets drunk one evening, lots of alcohol in blood, lots of alcohol and metabolites in the testes, and probably germ cells, including sperm cells, may be affected at the time. But then once spermatogenesis, continues to produce, the cells which were present in testes and sperm cells and all the prior stages present in his body at that time may be exposed. But once they have all been cleared, and the effect should not be there anymore unless the progenitor cells themselves have been affected. But the progenitor cells, they are protected in testes. There is a blood testis barrier which protects these germ cells. So high level of alcohol for just a couple of hours is not going to be enough. I don't know. May not be enough to affect germ cells, the progenitor cells. We don't really know, actually. No one has really looked at that in human, because in human you cannot collect. I mean, we do that in mice, but we need to sacrifice the mice to collect these cells. In human, progenitor cells, they are called spermatogonia stem cells. They are in testes. They are embedded in testes. I mean, you would need to extract the testes to look at them.
Okay. Can't do that. So can you describe your work with the mice?
So we focus on early life experiences, so postnatal, after birth, during the first couple of weeks of birth, and we are interested in the effect of traumatic experiences during that time. So it's a critical time window for human and mice, because the body is in development, and many of the cellular barriers, I mentioned the blood-testis barrier, but there is also a blood-brain barrier. Many of these barriers of the tissues are not completely formed, and things are still undergoing development. So for some aspects, it's a critical window of susceptibility. So we expose mouse pups, just newborns, to stress, and it's chronic stress. It's unpredictable maternal separation combined with unpredictable maternal stress each day, at any time during the day, for 2 weeks. So 14 days. And this is a lot. It's chronic, because a mouse pup is weaned. So it can be, doesn't need the mother anymore after 3 weeks. So it's 2/3 of the early childhood, if you want.
Right.
This would correspond in humans to from age 0 to age, I would say 11, 12, maybe.
Okay.
And it's a chronic stress. It's every day. And then these pups, after they have been stressed, they are 1 week after the stress. They are 1 week with their mother, with no disturbances, no stress, no nothing. So a quiet week in a way. And then so they are weaned, and then we wait until they are adult. So there are males and females, of course, in litters, and we test their behavior, their physiology, their metabolism. We do many analyses, and we found that they are depressed. They have increased risk taking. They are antisocial. They have problems of memory. They have problems, cardiovascular problems, immunological problems. So many things are altered in these animals. And we see that through the fathers, when these males, which were themselves exposed to the stress when they were young, if they get an offspring, the offspring will have very comparable phenotypes, symptoms. And if we take this offspring, breed it again, I get a grand offspring, again we'll see symptoms. And we did this to the fifth generation, and still in the fifth.
Wow.
There are some symptoms, not all of them, but risk-taking is one of the symptoms which is still observed in the fifth generation. Now, if we take females, the little females, so the sisters of these males that we bred till the fifth generation, we bred them once. So we went to the second generation. We did not go further. And we saw also that the second generation has symptoms similar to the females which were directly exposed themselves. So all of these indicates that there is indeed perpetuation of the effect of trauma, at least in the patriline. We can firmly say this in the patriline. In the matriline, we don't know because we did not go further enough, because you realize a mouse pup, like a boy, is exposed to trauma or whatever. His cells, including his future sperm cells, are affected, are directly exposed in a way. So it's not too surprising that his children may have effects, right? So if now we want really to talk about transgenerational transmission, we need to go one generation further. The generation when not a single cell, not even the germ cell that gave rise to this individual, the sperm cell that gave rise to this individual, has never been exposed at any point in time to exposure. And in females, it's the same. You imagine an expecting mother. So the mother, her baby, the germ cells in the baby are affected. So which means her child and the child of her child. So 3 generations in one lady are exposed. And indeed, in the US, there is really a high surge of autistic spectrum disorder. And many people think that it's linked to endocrine disruptors, in particular cocktails of drugs which were given to women to avoid miscarriage. That was very prominent in the US because these drugs, I mean, Americans have been eating medications a lot more than Europeans since a long time, longer than here.
Yeah.
And now you see many more cases of autism in the grandchildren of these ladies in the 60s, 50s, 60s, 70s who had been taking these medications. So it's one of the examples.
Is anyone directly studying that?
Well, there is a lady from California, Jill Escher. Have you heard of Jill Escher? She's a lawyer by training, but herself, she's born in 1965, and her mother took some of these medications, and she was very struck. She's a very, very smart lady. She has 3 children. The 3 are autistic, 2 very severely. Oh. 1 is okay, manages his life on his own. So she really dedicated her time, her life to dig into this. And she has collected lots of information about families in the US who have been exposed, mothers who have been exposed to that medication, but also to anesthetics. She has explained it. She's not a biologist, she's not a researcher, but she has been really outstanding in collecting data. It's epidemiological data. So now, of course, there are studies which are done in humans and in animals. In humans, it's only correlative. You cannot prove that's really the cause of the symptoms in the children or grandchildren. In mice, we can, because we can exclude many confounding factors. In terms of trauma, this has been done. I mean, our work really shows that it is in the germline. Endocrine disruptors, there is a very strong proof as well. High fat diet, low protein diet, the same. Now, of course, we would need to demonstrate the causality in humans. This will not be possible at all because of confounding factors again, but the status of the research at the moment is really suggesting really strongly that this is the case in humans as well.
Wow. So just to say that a little bit differently and to state the obvious, the difference is in the mouse pups, you can control for all the other factors. So you know that it's directly the trauma that's causing the consequences, for future generations. But in humans, obviously, there's too much going on. You live an entire life. You're affected by what's going on around you. And of course, we can't put a child in a lab and not have them, experience any of the rest of life. So that's sort of what you're describing.
Exactly. There is the fact that we control everything and that these animals, it's the only experience they have. They don't have any other experience. Not only we control, but it's the only experience. Everything remains the same. And also because we can use strategies. For instance, these pups which have been so the progeny of individuals of mice which have been exposed to stress, we can take the pups and give them to a normal mother. So a mother which has not normal, to a naive mother, a mother which has not been traumatized itself, to really check whether maternal care may be playing a role here. And this is, even if they are raised by a non-traumatized mother, we still see the phenotypes. We have done IVF. So we take the sperm of traumatized males and we produce an offspring without mating, without breeding, just by using the sperm. Right. Even if we take only the inside of sperm cells, the RNA, and we inject it into an embryo, one cell embryo, just control, and we see phenotypes in the offspring. So we've used multiple strategies to really prove that it's in the sperm cells that there are signals of a trauma which can be passed to the offspring and be responsible for symptoms.
Wow, that's fascinating. And obviously in humans, as you pointed out, you sometimes with mouse pups will give them to what you call the naive mother who hasn't experienced the trauma. Often in humans, the families continue to experience the consequences of the trauma or even new events that start to affect that. Yeah.
Yeah,
wow.
Absolutely. Because if you take intrafamilial violence, violence in families, I mean, usually it's associated with many different things: physical violence, verbal violence, alcohol abuse, neglect, humiliation, these kind of things.
I was going to ask you about the healing or the lessening of the consequences of the original trauma. And it sounds like from what you're describing that the consequences or symptoms or sort of, behavior lessens through the generations, but it can go through 5 generations. But is that accurate or is it still, I guess, do you notice a healing, I'm using that word very loosely, of the trauma through the generation or the consequences of the trauma through the generations?
There are symptoms which disappear in a way, but it all depends on the strength of the trauma. And unlike in humans, in our case, there are only 2 weeks of trauma, only 2 weeks. So the fifth generation, the ancestors were born 2 years before, even 3-4 years before. So there are no other possibility of or event which could revive the trauma, unlike in humans. So if we would use an even stronger trauma, have it longer, have it, adding different things, perhaps it would still affect individuals in the 6th or 7th generation. We don't know, so we cannot really make a firm statement about that.
I'm curious about kind of emerging technologies that are advancing your work. And maybe you want to talk about this a little bit. You do the biannual International Epigenetic Inheritance Symposium. Is that correct?
Indeed, yeah.
What sort of came out of that really intrigued you, excited you?
Well, the fact that there are more and more observations which confirm that exposure to whatever diet, a dietary insult, trauma, anesthetics, arsenic, many different exposures, they do have effects across generations. And the refinement of the phenotyping, so the refinement of the effects, and also more and more knowledge on the molecular correlates of that. What does it mean, for instance, to have a diabetic state in a mouse descendants of a male which has been treated with high-fat diet? What are the molecular alterations? And try to link the molecular alteration, let's say in liver, with those observed in sperm, because there is the phenotype and there is what causes the phenotype. And if indeed we assume that it's the sperm which has the signals and it fused with the oocyte, it gives an embryo which develops and develops. What is the link between these signals in sperm and, let's say, liver dysfunctions or memory problems in the offspring in adulthood? How does this unfold and gives rise to these symptoms? It's very difficult to understand that and to dissect out all the different steps. So there is more and more knowledge about what may happen. Another thing is that for paradigms which are not necessarily very strong, I mean, ours is super strong.
Mm-hmm.
So each time we see the phenotypes in the offspring, grand offspring, so on. There are many paradigms which are milder. And there is lots of variability in the effects, which does not mean that they should not be studied, because they may indeed be maybe closer to what's happening in humans. It's a little bit of this, a little bit of that, and that it accumulates, and that in some people it will show that, in others it will not show. So people are now starting to exploit or trying to extract information using novel statistical models to understand, to really model and have a conceptual framework to understand the different possibilities. What are the nuances that an exposure could bring in terms of symptoms, phenotypes, effects across generations?
I want to ask you about the term phenotypes. What exactly do you mean when you say that?
A phenotype is a trait. Let's say a behavioral phenotype, a metabolic phenotype, physiological phenotype. It's a function. It's a trait. I mean, you have brown hair. It's a phenotype. You have hypertension, or you or someone has hypertension.
Right, sure.
It's a phenotype. It's really a feature.
Okay.
And in mice, it's something that we can quantify and define qualitatively very precisely.
Where can people follow your work, see what you're doing, support what you're doing, look at your publications? Where should they go?
Oh, we have a website.
Okay.
Which is, I think, Mansuy Lab. I think if you Google my name, you find a...
Yeah. And we will link it too.
Yeah, oh, great. For support, I don't know, that would be great, because it's a really hard field of research, which still hits quite some resistance, because, the very classical view on heredity is that it's genetic. You are your genes, right? It's something different. We say that you are not only your genes, but you are what you experience. And you are also what your parents experience, and maybe what your grandparents experience, which is a completely different framework, conceptual framework for heredity. And there are still geneticists who resist, who don't want to hear that, even if there is very strong evidence, because it's like a dogma that heredity is genetic. It's a dogma, and even among the most intellectuals, big professors,
yes,
some of them are not prepared to reconsider that. So it's a field of research which is really, really hard. It's hard to publish. It's hard to raise funding for research. Everything is hard. Much harder than, people who use big microscopes and look into the brain and just report what they see.
Right.
There is no concept. There is no hypothesis. There is no risk in what they do, because, you just take something, observe and report, and that's it. So with all the financial restrictions which are ongoing, not just in the US, but also in Switzerland, even if Switzerland is super privileged for that. And if public funding agencies they have limited funds. Of course, they will choose to fund research that is pedestrian, that is secure, that does not raise any question, or that is guaranteed in terms of results. So we suffer a lot from that, unfortunately. But I think it's so important and so relevant for human beings. It's very important that people are aware of that.
My last question is, what do you hope that your work kind of gives us?
Yeah, that people understand the biology a bit better, and understand heredity. And for diseases like depression, depression, there is a lot of taboo. Many people are depressed. They don't want to admit that they are depressed, because in our society, being depressed means you failed your life, right? You are weak. You cannot resist. You are too weak.
Yeah.
A lot of guilt. That's why one of the reasons why many depressed people don't consult, because they don't want to admit for themselves, and they don't want to, there are rarely any coming out about depression. Now, if you consider that indeed there may be nothing wrong with yourself, with your life, with your choice in life, but that may come from your parents or even your grandparents. Yeah, just you inherited this shape of nose. There is nothing you can do. It's just genetics. But that it also may also be the case for every other function in your brain, including dysfunction in the brain, like depression, schizophrenia. So that can really help a lot. People understand where the condition may come from, and consult, and maybe that may be easier to fix, because if now you consider that depression is just like any other disease, just have to analyze it, see what are the symptoms, see what you can do to alleviate the symptoms, and life goes on, instead of saying, I feel guilty, I will never get out of that, and so on. I mean, colleagues, psychiatrists say that can make a very big difference, actually.
Wow.
Cardiovascular disease is a bit the same, because, we found, for instance, in our mouse model, that they have heart failure. And you see, wow, that's stress. They have not eaten anything bad. They don't smoke. They don't, they have normal activity. So what is the link? How come it was not known that indeed cardiovascular alterations in an animal could be due to the stress of the grandfather? So if cardiologists know that, you see, it can also change completely the attitude towards medicine, and maybe towards diagnostics, and understand the cause of disease. Because rather than treating just the consequences of disease, if you understand the cause, then that can really change approaches to medicine.
That's wonderful. Well, thank you so much for what you do, and then for talking with us today. So appreciative, Doctor.
Thank you. Thanks for your interest.
So excited you were here today. Thank you so much for listening. I would love to hear your story. If you're interested in sharing it, please go to justlikenana.com to submit your story. There are also resources there if you're interested in learning more about discovering your ancestry or breaking some trauma cycles in your family. I have an ask of you. If you enjoyed today's show, I would love if you subscribed and let a friend know they might enjoy it as well. Talk to you again next week. Can't wait. Just Like Nana's theme song was written and performed by Carter Penny.